You've probably learned or heard about DNA, but have you ever seen it? With the strawberry DNA experiment, we'll extract, isolate, and observe the DNA of a strawberry in a matter of minutes. Though it seems difficult to conduct, in reality it is pretty simple due to the special characteristics of strawberries. You don't have to be a scientist nor use complicated tools. It's easy, fun, and all you need are some household materials. DNA contains the genetic material determining the makeup of all organisms. There are obvious differences between plants and animals. At the chemical level, the cells of all plants and all animals contain double helix DNA and the same four chemical building blocks called nucleotides. What makes these plants and animals different are how these four nucleotides in DNA are arranged. It is their sequence which determines which proteins are made, and the information they encode decides the fate of the organism, whether to produce scales or leaves, legs or a stalk. To obtain a DNA from a cell, scientists typically rely on one of many DNA extraction kits available from biotechnology companies. Strawberries are chosen for this particular experiment because they yield way more DNA than other fruits, and they are octoploid, meaning they have 8 copies of each type of DNA chromosome. These special circumstances make strawberry DNA both easy to extract and to see. The materials needed for this experiment are rubbing alcohol, 3 strawberries, salt, water, dishwashing liquid, glass or small bowl, plastic Ziploc bag, measuring cup, and measuring spoons. Why do we need these materials? To extract the DNA, each component of the extraction mixture plays a part. Soap helps to dissolve cell membranes. Salt is added to release the DNA strands by breaking up protein chains that hold nucleic acids together. Finally, DNA is not soluble in isopropyl alcohol, especially when the alcohol is ice cold. Alcohol is added to cause the DNA to precipitate out. Create your extraction liquid by mixing 1/2 teaspoon of salt, 1/3 cup of water, and 1 tablespoon of dishwashing liquid in a glass or small bowl. Put the strawberries into a sealable Ziploc bag and push out the extra air. Seal the bag firmly. Smash the strawberries with your fingers for 2 minutes or blend with a blender. Add the extraction liquid to the strawberries in the bag. Pour the strawberry liquid from the bag into the funnel into a tall glass. Remove the filter once the glass is full, taking out the wet pulp remains. Pour 1/2 cup of cold rubbing alcohol. Wait for 7 to 10 minutes for strawberry DNA to appear in the form of white streaky stuff. Dip a spoon into the extraction to take out the gooey strawberry DNA and place it in a bowl. Wait for 24 hours for the DNA to harden. When you added the salt and detergent mixture to the smashed strawberries, the detergent helped pop open the strawberry cells, whereas the salt helped create an environment where the different DNA strands could gather and clump, making it easier for you to see them. Upon adding the salt and detergent mixture, bubbles were visible in the bag because of the detergent. After adding the cold rubbing alcohol to the filtered strawberry liquid, the alcohol precipitated the DNA out of the liquid while the rest of the liquid remained in solution. There was white, clear, gooey DNA strands in the alcohol layer as well as between the 2 layers. A single strand of DNA is extremely tiny, too tiny to see with the naked eye. But because the DNA clumped in this activity, we were able to see just how much of it 3 strawberries have when all of their octoploid cells are combined.